Thanks for coming. This webinar today is going to be a very general overview on Anuran reproduction and control. And it's basically sort of laying the groundwork for some of the basic reproductive strategies that animals in this taxonomic group employ. And hopefully we'll start to give you a little bit of a background regarding where the rest of the webinar series will go in terms of the application of arts and other reproductive technologies for captive amphibians. So without further ado, I'll let Gina take it away.
Thank you, Nat. Okay, so welcome everyone. Thanks for joining us today. We'll talk a little bit about reproductive strategies. And okay, I'll start. So the order Anuran displays an enormous variety of reproductive strategies compared to other orders or even entire taxonomic classes, sorry. So the reproductive physiology and reproductive strategies are usually tightly related, and it is important to become familiar with both to be able to develop the most appropriate arts protocols for your species. Usually, strategies vary taxonomically, geographically, and due to abiotic factors. Reproduction in Anurans is also controlled by endogenous factors or neuroendocrine factors that control gamete growth and maturation and reproductive behavior, and also environmental factors. Seasonally variable environmental factors regulate the different stages of gametogenesis, which is the production of gametes, and this involves proliferation, growth, and differentiation, also maturation of gametes and breeding. The most important or common factors that interfere with or affect reproductive strategies are temperature, photoperiod, food availability, seasonal rainfall or humidity, among others. For tropical, subtropical, and desert species, seasonal rainfall and humidity is the most important or relevant factor. And for temperate species, usually it's temperature, although there is evidence that photoperiod is also an important factor for these species. So reproductive strategies for each species have evolved with the aim to accomplish maximum survivorship of eggs and larvae, minimum physiological stress, facing fewer predators, maximum food availability for hatchlings, best egg incubation conditions, and suitable environments for hatchlings to thrive. And this results in different strategies for many species on this order. Okay, so many Anuran species, either temperate, tropical, or subtropical, show seasonal reproductive patterns or activity. And we refer to it as reproductive cycles. So what are these? Usually it's the length between two breeding seasons. Most Anurans breed seasonally once a year. And these cycles can be either discontinuous or cyclic, which are the seasonal species, potentially continuous or continuous, which are acyclic, and the species reproduce continuously throughout the year. The species with a single breeding period usually show changes, physiological changes and histological changes in the ovarian testes. The relationship between gonadal activity and sexual behavior, and we refer to gonadal activity as the secretion of steroids, gonadal steroids or hormones, and spermatogenesis, can be shown in three different patterns. The species that show an associated reproductive pattern are usually the ones where the gonadal activity coincides with the onset of the sexual behavior. Those showing this disassociated reproductive pattern is where the gonadal activity is independent of the onset of the sexual behavior, or not always coincides with that, the sexual behavior. And the opportunistic reproductive patterns, which are species that are quote unquote ready to go, and usually it's an external factor such as sudden drop in temperature or a rainfall event that will prompt the breeding event. Usually, tracking and predicting the reproductive cycle can be accomplished through the calculation of the gonadosomatic index, or the GSI index. But this requires euthanasia of animals throughout the year. And basically the calculation is dividing the gonad weight between the body weight times 100. The highest numbers are usually gotten prior to mating, but this is not recommended for threatened species or captive species under conservation programs. We recommend alternative methods such as ultrasound, basically most suitable for females and following throughout the year. So depending on the region, some species show, let's say, an average pattern. Temperate species, usually most of them, show a discontinuous or cyclic or seasonal reproductive pattern. And the main factor affecting these species is usually temperature. For tropical species, we'll find species with discontinuous and continuous patterns, and rainfall is the most important factor for this.
Thank you. This figure illustrates very broadly what we're talking about with regards to breeding strategies. Seasonal or discontinuous groups and versus the continuous breeders exhibit differential strategies in response to the environments that they live in and the pressures that they must adapt to. For example, in the case of seasonal breeders, typically their environments are characterized by short active periods in which they must do two things: breed and forage. During what is considered a short and a highly active period of time before the onset of winter, before diving back into an inactive period known as, in the case of your cold temperate species, brumation, also known as overwintering, or estivation, which is usually undergone by animals that live in hot desert environments in order to avoid desiccation. Now, as altitude increases or latitude increases and decreases, these different species would choose these alternative survival strategies in order to withstand the extreme environmental pressures such as drought and the winters that I mentioned. But this means that their breeding must be in tune to these extreme response in an extreme way to respond to the environments that they live in. So some of the desert dwelling species that estivate, for example, might burrow into the ground, sometimes for years, waiting for that perfect rain downpour to come, and then they come out and they breed explosively. So as you can see at the far right end, that's what I mean by the rain. It can either be an increase in moisture in the ground, or even the vibrations of the raindrops as the rains start to come down on the ground that actually elicit the emergence of these animals. And these processes usually only last a few days, and then the animals go back underground and wait the next rain event. With cold dwelling species, environmental cycles may be a little bit more cyclically reliable, occurring at least once a year in response to winter. They can breed. So these are what we call, they're designated the pervasive breeders, as they use, and their breeding periods last a few days or weeks. Tropical species can continuously breed year round, but this doesn't mean that they're not seasonal. It simply reflects the more consistent environmental conditions that they live in, which allows for breeding strategies to occur throughout the year, and may also allow for more than one clutch in the case of females to be spawned per season. So relating to the environment in which the animal lives, we can broadly categorize the resulting breeding strategies. It can be explosive, pervasive, or year-round. And importantly, though, we should note that even within a species, there can be intraspecific variations between the populations of that species, depending on where their distribution lies. So a species that's widely or globally distributed and quite common may actually occupy habitats at different elevations and different latitudes, which means that the timing and the duration and the environmental influence on their breeding strategies may work slightly differently. And this is obviously going to become a bit more apparent later on in the talk as we kind of disclose more about ovarian and gamete control in general, because you need to be aware for your captive population where it is that, geographically speaking, that you obtained your animals from when you bring them into captivity and you set about breeding, and understanding what their breeding strategies are like. The mechanisms by which these pressures exert their influence on the actual physiological systems is very complex, and so I'm just going to briefly go through this to give you an idea of how this all works. And basically, the abiotic processes that affect an organism's physiological adaptation to the environment, such as temperature and photoperiod, will do this. In this case, temperature and photoperiod signals can be conveyed through the eyes, and they affect photoreceptors in a similar way. Temperature affects the changes to photoreceptors in the same way that light does. And last week, Dr. Trudeau talked about the endocrinological mechanisms that drive reproduction. So briefly here, if we take the blue colored boxes representing the cooler and shorter days as an environmental cue, and the yellow is the warmer, longer days, typically associated with summer. And then it follows that these differences in temperature and photoperiod affect the secretion of these same hormones that Vance spoke about to inhibit or stimulate reproductive processes in males and females, such that the gamete production coincides with the appropriate time for reproduction in the environment. Genetic control and hormones act in conjunction to control these processes, and genetically speaking, fast changes through epigenetic modifications allow quick adjustment of gene expression. And what this means is that it gives the amphibian the flexibility to respond quickly to environmental changes and be able to sort of determine physiologically when the best, most appropriate time to breed is. Regardless of the mode of reproduction, this means rapid and reversible implementation of physiological and structural changes to the organism's tissues, and includes structural modifications as well as biochemical and metabolic changes to tissues and cells. It is these modifications at organ and cellular level that allow gonadal and gamete development to proceed. Back to you, Gina.
Thank you. Okay, so Anuran showed different spawning strategies, among which we'll find oviparity, which the fertilized egg and embryo develops outside the mother's body. Most Anuran species are oviparous, and they show free-living larvae and no parental care. We also find ovoviviparity. The fertilized egg and embryo, in this case, develop inside the mother's body. The embryo and larva will feed on the egg yolk, and eggs are usually retained in special cavities, ectopic sites, or such as the oviduct, or a special chamber on the stomach in some species. And we have viviparity, where there is no egg and the embryonic development takes place inside the mother's body. Today, there's also one species. There's only one species known, the Western Nimba or Mount Nimba viviparous toad for the Anuran order. And we also have the oviposition strategy or nesting site selection for tropical and subtropical species. There's been a tendency to move away from water to avoid predators. Nevertheless, we find many species that still reproduce in water. But there is also a wide variety of strategies, such as the leaf frogs. They will lay the eggs on leaves above bodies of water, and then the tadpoles will hatch and drop into the water. And then we'll have the species that build the foam nest, is either on the ground or in, for example, tree branches. The species that will construct burrows at the edge of water or farther away from water to protect their eggs from predators. They will lay eggs also in tree holes full of water, or in bromeliads, for example. These are some examples, and you can see here, for example, marsupial. They're also terrestrial, considered terrestrial, and different ways to lay the eggs in the water. There are many oviposition strategies for Anurans. Nevertheless, external aquatic fertilization without parental care keeps being the most widespread strategy, and it's the ancestral strategy. And this is most common strategy in temperate frogs. Okay, so let's go a little more into detail with the male reproductive cycle. So those of you that attended Vance's webinar, the last webinar that was given, he went into detail on the hormonal control for reproduction on Anurans. I will just give you a quick overview. And we know that Anuran control, reproductive control, is under the hypothalamic pituitary gonadal axis, known as the HPG axis. Basically, this is involved in the regulation of reproduction, controls reproductive behavior, the production of sex hormones. Sex hormones promote gamete proliferation and differentiation. And this axis is mostly triggered by environmental factors, and it's a major regulator of spermatogenesis and ovogenesis. And basically, we have the hypothalamus that produces the gonadotropin releasing hormone, GnRH, that acts on the anterior part of the pituitary and promotes the release of luteinizing hormone and follicle stimulating hormone. These two hormones travel through the bloodstream, and they will act on the gonads and promote the release of sex hormones. And then the process of spermatogenesis is triggered. A quick overview again, back to cell biology 101. We have the germinal cells on the testis, the spermatogonium, that will go through several different divisions, mitotic and meiotic divisions, and basically will produce a primary spermatocyte, two secondary spermatocytes, 4 spermatids, and at the end 4 sperm cells, mature sperm cells. So this is important. Why? In the application of arts. There are several factors that you need to understand of yours or your own species. First, in general, spermatozoa are inactive or not motile in the testis, and motility or activation is dependent on osmolarity. Testicular osmolarity is usually around 300 millismol per kilogram, and sperm activation occurs when the sperm comes in contact with the urine, and osmolarity drops below more or less 150 millismol per kilogram. So sperm in Anurans is released as a spermic urine. It's a mix of urine and sperm. Activation or motility consumes most of the cellular energy of the spermatozoa, and duration of motility, it's dependent on this, let's say, this energy that the cell holds. This will vary among species. Not all species present the same duration of motility. And it's important for the development of arts, especially for artificial fertilization or cryopreservation protocols, because you don't want to freeze sperm that's already dead, it has already consumed all the energy, or create osmotic shock on your sample and kill all your cells. So previous studies on the characterization of the sperm of your species needs to be done before proceeding with the development of arts. So the Anuran testis, basic characteristics, these are paired ovoid organs. Basically, they're formed by folded seminiferous tubules, and they're surrounded by connective tissue. And the germ epithelium or testicular parenchyma is arranged in seminiferous locules. You can see them here on this picture that I'm showing you here. These are the spermatogenetic cysts. Basically, these are clusters of cells associated with or 2 Sertoli cells. There are 2 main types of cells in the testes that are affected by sex hormones, which are Leydig cells and Sertoli cells. All these cells in these clusters are in the same differentiation stage, and this is called synchronous sperm development in Anurans. And you'll find different clusters in different differentiation stages, but all the cells inside that same cluster are in the same differentiation stage. Then at the boundary of the seminiferous locules, you'll find the spermatogonia that we saw on the previous slide. Towards the middle, you'll find spermatocytes and spermatids, and towards the lumen of the locule, you'll find the mature spermatozoa that's ready to be released. So it's important to understand the morphology and cell cycles of your own species, which are not the same for all species, of course, usually species specific, for again, the development of arcs. And testicular assessments or histological assessments can be done from tissues collected from carcasses, fresh carcasses, we recommend. So sperm characterization and morphology. These are some pictures of Panamanian golden frog spermatozoa. Some factors or characteristics that we recommend to assess are sperm morphology, sperm concentration, percentage motility, percentage forward progressive motility, spermicute osmolality, and spermicute pH. After you have assessed all these parameters, then you'll have a general characterization of the spermatozoa or sperm samples of your own species. In this case, for example, we see a normal cell, sperm cells from, from Panamanian golden frog. And we see different structures in this cell. We'll see a very small acrosome. The acrosome is the structure where the special enzymes that they digest the egg yolk and make fertilization possible are located. Then we see a head and a filiform head. Then we see a structure called the mitochondrial vesicle, which is filled with mitochondria. And then in this case, we see a long tail with an undulating membrane and two filaments. There's also important the characterization of abnormalities in your samples. In captive species, for example, you'll have individuals with best quality sperm than others. And it's really important to determine which ones are those, because those are the ones that you will basically want to breed. Some examples of abnormalities, you'll find, for example, sperm cells without an acrosome. This cell can be motile, but cannot fertilize an egg. This is a bicephalic head, which is a pretty serious abnormality, or abnormalities in the tail. In this case, for example, there's a coiled tail here and a broken tail here, which makes impossible motility for these cells in particular. Motility then can be assessed using either manual assessment through the use of microscope and a hemocytometer, or using automatic systems such as CASA system, which are expensive, but really, really good because they will give you exact numbers. We know that manual assessments, they usually have an error margin that is higher than automatic systems. Other morphological assessments are, for example, the use of Hodge staining and propidium iodide that will give you an idea of the size of the nucleus compared to that of the head. And the use of staining such as mitotracker, which will bind to the respiring mitochondria and indicate where the mitochondria are located in the sperm cell of your species, and the amount of respiring mitochondria or the activity. In this case, for example, the mitochondrial vesicle is not completely round. It's kind of elongated here. So you can see that it's showing brighter green where the mitochondrial vesicle is located here. So in this species, this accessory structure, it's filled with mitochondria indeed. Another interesting factor in these three examples, we have three species here: Tripion spinosus, This species reproduces in 3 holes full of water. This is static water. This is Panamanian golden frog that reproduces in streams with current. So the sperm needs to swim many times against the current. And we have Craugastor evanesco, which is a terrestrial species. The female burrows the eggs in the sand, and the male will urinate on top of the eggs. So for Tripium spinosus, we have a fairly big head and a single cell tail, single filament tail, sorry. And the movement is pretty slow. In the case of Panamanian golden frog, we have an undulating membrane wider, and the wavelength it's bigger because they need to swim very strongly against the current. And in the case of Craugastor evanesco, we have a sperm cell that has a really, really long head, but the tail, in proportion to the head, is super long. It also has an undulating membrane. It's thinner than that of those that swim in the current, and the wavelength is really short. They move really, really slow compared, for example, to Panamanian golden frog sperm. So it's really interesting comparing the morphological characteristics of the sperm with the reproductive strategies of the species. And this helps us understand and better develop artificial reproductive strategies for our own species in captivity. Nat, your turn.
Thanks, Gina. Okay, so now we're going to shift really quickly to the female side of things. Gina, if you can, yeah, thank you. So I'm going to kind of continue on that vein a little bit, more of talking about like the seasonality of things, and I will progress through the talk and give you an example of one of the species that I have worked on very closely in the last few years. So as I mentioned before, with temperature changes being one of the direct triggers for physiological transitions from inactive periods into breeding and foraging, females, particularly high altitude temperate species like the species I worked on, which is the mountain yellow-legged frog, has their reproduction, specifically folliculogenesis, so the development of follicles within the ovary, is tightly linked to temperature and environmental changes in temperature. So, and this is characterized by a series of cellular events that take place in the nucleus of the cytoplasm of the developing oocytes, and these are key to the growth and maturation of these oocytes. Now, exposure to the environmental changes that we've already spoken about dictate that these cellular changes through hormonal and genetic control will occur through hormone and genetic control. So in a brood-mating species, for example, like the mountain yellow-legged frog, the growth and the accumulation of yolk, otherwise known as vitellogenesis, and the nucleocytoplasmic maturation often occur during the time which these animals are actually inactive, so during overwintering periods. And the fat that they've accumulated over their active foraging periods is the resource that will actually be poured into the growth and maturation of these eggs in order to be able to come out and breed the following year. And mostly the fat that females accumulate during foraging actually is more tightly linked to an investment in reproductive strategy than it is in actually surviving the cold winters, because of the metabolic changes that occur in the females during these periods. For example, they will actually not require a high amount of calories to survive, because metabolically speaking, they've become baseline inactive. And so really what they do is they take the fat stores that they've derived from there and they pour them into this vitellogenic process. And then that then pushes the eggs through maturation in order to come out and breed the next year. So typically folliculogenesis occurs asynchronously in the species, in a very species specific manner, and the variability between species is going to be incredibly high. much like Gina sort of addressed with the males, this really has to be looked at on a species-specific level. So the developmental timing, the size, and the number of the eggs will correlate to the environments in which they're typically laid in, and will sort of those evolutionary strategies will be tightly linked to how to secure the development and the survival of these eggs once they're fertilized and proceeding through embryonic development. Broadly speaking, there were 3 stages of development, which is pre-vitellogenesis. And if you look at the diagram, your top left-hand corner, these images are taken from an Australian species known as Latoria phalanx, which is the eastern dwarf tree frog, and as a more common species that we use at University of Newcastle as one of our models. And so in the top left-hand corner, you can see is female, it's ovaries that are in the early stages of pre-vitellogenesis. So this is primary and primordial and primary follicles, which are pretty much devoid of any yolk. So sitting in that first stage of development. If you move to the right next to that, then you'll actually see how there's started to be an accumulation of yolk in the eggs, and that's that kind of yellow viscous color. And that's sort of an intermediary phase, like I said, what we call vitellogenesis. And then if you look down, the 2 diagrams below are representing your final stages of maturation. And what I meant by asynchronous is that not every single follicle in the egg is going to be ready for ovulation at every breeding season. And this again will be species specific, but as a whole, generally, a lot of the species, at least that I've worked with, seem to have this asynchronous development. So they don't develop every single follicle in one go, but they will have a high number of follicles ready to go. In this species, you can also see the difference between the animal and the vegetable pole, which is the dark and the lighter side of the egg. In the Mount Yellow-legged frog, for example, you won't be able to distinguish that, and the eggs are a uniform color. So this is, in this species, in Latoria phalanx, this is actually quite a useful way of telling how mature your eggs are and whether they're ready to go. Next slide, thanks. Excellent. So in telling you that, all of those things, all of those stages that are linked to your seasonal cycles in the environment and should coincide with the female being ready to lay, coming out of brumation. Now, in this slide, it's data that was collected a few years ago by myself and the staff at the San Diego Zoo when working on the Mount Yellow-legged frog. And basically what had transpired with this population was that it was set up as a salvage operation and egg clutches were brought into captivity, and animals were metamorphed in captivity and set up as a breeding colony. And for the first few years, were not brumated appropriately because there was some nervousness regarding the application of these artificial brumations to the animals in case of increased mortality. But as the years progressed, we were able to convince our partners to let us put the animals through brumation. And what we saw across the years was definitely an increase in the number of females that started laying eggs, an increase in the number of eggs per clutch across the years, and an increase in survival to tadpole or hatching, and even through metamorphosis as that brumation, as those brumation periods were brought down to temperatures that were more equivalent to what would be observed in the wild. For longer periods of time. In this case, now typically the animals are put through about a 3-month brumation period from about December till March. And this seemed to help under natural conditions, devoid of any kind of artificial technology intervention, to increase increase, obviously, the number of animals produced in the colony, and across the years, we were able to begin reintroducing more and more metamorphs into back into the environment. What we did in order to be able to sort of begin to ascertain a little bit more information as to how these things were progressing and how we would be able to sort of make better management decisions on who, when, and what to breed every year, we incorporated the use of ultrasound. And so if you look, and we ultrasounded during brumation. In this first slide, I'll show you some more images later. But in this first slide, it basically was characterizing what the ovary looked like throughout brumation in the first month, in the first, second, and third month. And I just want to note that even though in this slide you can see these images sort of characterized through the first and second, third month of brumation, females within the species will vary largely as to what stage of ovarian development they're in when they come out of brumation, and I'll talk about that in a minute. But broadly speaking, what you have with a typical female is you see the small follicular dots, which we call echogenic dots. And if you look at image A and image B, those are characterized by by the arrows there, the little white dots. And that's typically what you see when follicles are starting to accumulate that yolk. So when they're going through vitellogenesis. If you go into the second month of brumation, you can see those echogenic dots becoming even bigger, and those darker spots, again, looking at signifying the growth of the actual follicles and the further accumulation of yolk. And then as you get through to the third month of brumation, you can actually see the eggs fully developed. On the other side of that, I included an image of what an ovary looks like after spawning. And you can see basically what you've gone back to is this sort of gray haze. And that's because whatever follicles are left in the ovary at that time are devoid, or you've gone back to your primary follicles and they're devoid of yolk. So you're not going to pick up those echogenic dots in the same way. Next slide, thanks. So the other thing to have in mind, again, is also looking back at the natural histories of your animals. It's important to know that you can only understand so much about what's going on in ovarian control if you also look at the behavior of these animals. And when I started at San Diego, typically these animals were paired together. They were assigned pairs, and they were allowed to breed for about a week to 10 days before they were separated again, and then males and females were kept separately throughout the rest of the year and just allowed to sort of be in those in separate tanks. What I decided to do was that, given that there was very little information about how these animals bred in the wild and how long they bred for, was to spend the first 2 weeks of my first year there in 2014 basically just voyeuristically watching these animals morning, noon, and night. And that involved me being in the lab 24/7 and basically just watching them and taking down certain behaviors that I thought looked like something worth noting. And what that led me to was basically, broadly speaking, characterizing 3 different stages of reproductive behavior. And that's passive amplexing, which usually occurs within the first couple of hours of putting males and females together. The males and females will get paired up. The male will start amplexing a female, and the female will then move around the tank with a male just strapped to her back, essentially. Later on, that amplexing becomes a little more active, and by what I mean by that is that the male starts to exhibit certain behaviors. In this species, we don't hear, they don't call as such, and so there's some evidence that they make underwater noises, but they don't actually have that typical calling that's associated with a lot of other Anuran species. But what I did see was the males start to actually squeeze quite tightly on the female and rub their abdomens with their back legs. So that I sort of characterize as an active amplexing. And then to the final stages, which then seem to correlate with the onset of spawning, was the female started to look for things to stand up, do handstands up against. And the reason for that is because, as I would discover later, once the female is ready to lay her eggs, she needs to find hard surfaces to attach her eggs to. And so a lot of those handstands were related to pushing those eggs up and sticking them up against a rock surface or a hard surface, at which point, hopefully the male's also urinating sperm on it and actively fertilizing those eggs. So if we go down and we look at the ultrasounds, which is some of what I was talking about in the last slide, and we look at the different stages, I start at if you look at the right hand, again, you go back to that first no kind of empty, what we'll call the empty ovary, post-breeding, and I've called it stage 1 just because it's the beginning and the end. But basically stage 2 is your early follicular genesis, and stage 2 through 4, females can come out of brumation at any one of these 3 stages. And all that means is that typically, if you come out of brumation at a stage 2 and you're amplexed by a male, you're going to take longer to spawn than if you come out already at stage 4. And this was important because in actual fact, after many, many hours of watching these animals, what came to light was that they could spawn anywhere from 24 hours to 5 weeks later. And so separating these animals before that time meant that we actually weren't capitalizing on our breeding strategy at all, because some females were just not quite ready to go. And there was something about being paired up with those males that pushed their ovarian development ahead, whereas other females were already ready to go when they came out of brumation. And what that variability is related to, I have no idea. Next slide. So the other thing that we now can start incorporating is other reproductive technologies that can help assess us. This is typically, particularly in zoos, widely used for other taxonomic groups, especially in mammals, and that's consistent monitoring of hormone levels through the annual cycles of primarily females, but sometimes also males. And we can also do this with amphibians. It hasn't typically been done a lot, and it's certainly not common practice within zoological institutions to monitor our herps in general, either amphibians or reptiles, but it is possible. And what I was lucky enough to do in my first postdoc at Mississippi State was actually pair up with some of the folks at the Mississippi State Chem Lab and start developing protocols of mass spectrometry to monitor these hormones. And this we've done, we're developing over a number of matrices. One is urine as the most non-invasive matrix that we could come up with at the time. We're now looking at using saliva as well and skin secretions. But we've also looked at trying to monitor hormones in blood samples. So that's extracting serum and the reason we want to use mass spectrometry is because obviously getting some of these matrices from these animals is quite hard, and you're only going to get small volumes for every sample. And what mass spectrometry does in comparison to your traditional assay methods is it actually allows you to screen multiple hormones per sample at a time. So whilst the methodology is a little bit more expensive, in the long run, what you have is the ability to screen over 90 samples at any given time, and with our protocols up to 7 hormones per sample. So obviously this is very handy, particularly given that sometimes you only come out with volumes in the low microliter kind of scenario. Next slide. Thanks. So as I say, we've been developing some mass spectrometry, and what this allows you to do is take these very small volumes of whatever matrix you develop your protocol for and shoot it through this mass spectrometry, which essentially divides and breaks up all of the particles in your sample, and then you can actually, quantify independently each hormone within one sample, and that's why it's so handy, especially in the case of amphibians. And what we do then is correlate these hormone levels back to our ultrasounds, and we can look back through our breeding history and sort of go through the individuals and the species as a whole to look at the different breeding histories for our animals. And this is obviously important in terms of, not only in terms of natural breeding, but when you're thinking about the application of assisted reproductive technologies, it's important to know where your animals are physiologically before you start injecting them with hormones. And unfortunately, in the past, given captive breeding programs are often under pressure to breed these animals because a lot of the species that we work with are either critically endangered, and there is some sense of urgency with having to produce animals, but we don't always have the appropriate background information as to when the most appropriate time to inject animals is, because we haven't actually done all of this sort of platform groundwork beforehand. Next slide. So some of the key features for inducing females with hormones is to understand, oh no, technical glitch. You're killing me.
Sorry, hold on. I don't know what happened. I'll be back.
I'll take a break here. And I had you all captivated at the edge of your seats. If anybody needs a bathroom break, this is the time.
Okay, it's back.
Is it?
Yeah, on the other screen. Let me get you back where you were.
All right. Thanks. There we go.
Oh, hold on.
Thank you.
There you go.
Excellent. Thank you. So anyway, like I said, some of the key features of understanding hormone induction in females is obviously, like I just said, is to have a little bit of knowledge about what the breeding history of your animals is. If you've never worked with these animals in captivity, then chances are you need to collect some of this information before you decide to go down the arts road. Female anurans are typically a little bit more difficult to induce hormonally compared to males, and this is very variable across species and even genuses. For example, in Australia, we still have a lot of trouble with inducing ovulation in tree frogs, whereas some of our more terrestrial ground-dwelling species have been induced quite successfully. So it's important to understand that the species are going to respond differently to this. Sometimes females require a low dose of priming prior to administering a higher dose, or what we call an ovulatory dose. And sometimes you'll require 1 or 2 lower dose primings 1 or 2 days before. It depends. Again, this is all part of developing individual protocols for each species. And sometimes you may actually have to give multiple hormone treatments depending. I mean, if the ovary is not responding to the environmental cues that you're administrating in captivity, then perhaps you're going to have to build that hormonal drive with exogenous induction, but you're not going to just get it done in one go. You might have to actually trial some multiple doses across time. Next slide. So after the first year of 2014 of breeding the Mount Yellow-legged frogs, naturally we still had a little bit of doubt as to whether we were maximizing our output, and so we did trial Amphiplex, which is that GnRH metoclopramide combination that Vance talked about last week. And in the case of these animals, we didn't have much success with that. We only trialed it over the one breeding season. And as you can see from the graphs, there was really no significant difference between the controls seen in blue versus the hormone treatment. There was an incredible amount of variability, of course, but overall, statistically, it didn't seem like the number of eggs that were laid by the controls or the hormone-treated animals were too different, and certainly the number of embryos cleaved from each clutch was not different. And so that year we determined that probably it hadn't worked, but not necessarily because of the hormone. I mean, the hormone protocol might obviously needs a lot of work, but also because at the time we were going through a transition in the lab where we were also tweaking a whole bunch of other husbandry related factors, such as manipulating light and temperature and changing the diets of the animals. And so there was a lot of confounding things that probably require readdressing in terms of how these hormone protocols work for these animals. But certainly in this instance, it didn't seem to have much effect. And there's ongoing work in this lab continuing around this. Next. So just to give you an idea, this is back, there's a little video up here on the left. This is that pair that was featured in a photograph up the top as well. And it kind of shows, Gina, can you, yeah. It sort of shows some of the behavior of the female right there as she's begun laying eggs. You can see the male's like sort of squeezing her and moving around. And she's sort of in that very characteristic pose of a handstand, where she's trying to find places to continue laying and sticking her eggs. And so this is that torch that you can see. That was me probably around 2 or 3 o'clock in the morning with my head torch watching these animals in the lab. On your right is a table, and I just wanted to feature really quickly that the other thing to keep in mind with these females is that because there's such an enormous energetic investment in actually producing eggs, it should be noted that when you're trying to breed species in captivity, there is a chance that your females may not be ready to lay every year. Just like some species in the tropics can breed multiple clutches a year, and others may actually skip breeding seasons. And the Mount Yelliga frog certainly seems to be one of these species. And that has changed over time as husbandry practices have also changed. And so maybe there was something remiss in the husbandry practices before, but certainly females, when I began, seem to have no more than 3 consecutive breeding seasons in them before they'd take a break of at least 1 or 2 years and then come back and breed again. So that's important to note, because if you're thinking that your female hasn't bred that year and you've decided to shoot her up with hormones, but maybe she's just taking a break, you might want to think about it twice and just give her a break, right? They're not factory. It's not a factory, and we're not trying to produce eggs every year as fast as we can. We're actually trying to get the best quality gametes to ensure the fittest offspring, particularly when you're talking about reintroduction programs. So in this table, this is also a characteristic of boreal toads, which I also worked with in Colorado. The Southern Rocky Mountain boreal toad is documented in the wild as skipping breeding seasons, and females can go up to 3 or 4 breeding seasons before they breed again. And so that's important. And again, it goes back to that energetic investment in breeding for females. Next. So when you want to apply arts, there may be times when, can you hit that video, Gina? Thank you. When you have a species, and here we have an example of a Southern Rocky Mountain boreal toad. If you look at her abdomen and you can see those contractions, this is something that I had picked up from the species after hormonal induction. These females will lay in the absence of a male. But what you see just as they start to lay eggs are these really, really vivid kind of abdominal movements where you can see, which, correlates with the eggs starting to come out. And what we did with these animals at a place called the Native Aquatic Restoration Facility in Alamosa, Colorado, was to actually, they weren't breeding very well together. So what we did was we induced the males and the females separately, and we were able to go in and do IVF. We were able to bring in sperm from the wild and mix it with female, with eggs from females in captivity to try and sort of balance out those genetics and incorporate some wild genetics back into the population. And so once you understand how all of these things sort of occur, even if it's on a very superficial level, you can actually start to incorporate your other assisted reproductive technologies like IVF to try and help you maximize that breeding in captivity if you're having some trouble with that sort of natural approach. Next. The last thing I'm going to talk about is a paper that Leah Jacobs et al. Just put out in our recent Reproductive Fertility and Development Amphibian Special Edition. And it's a really good short communication on correlating the use of hormonal monitoring in females and males with what we started to observe a few years ago, which was some of our females from the founding population, which were older, they were about 12 years old at the time, 11, 12, started developing these characteristics, male characteristics, and they started growing nuptial pads, which is something that's very clearly a sexually dimorphic male characteristic in many species, and is associated with testosterone levels, like the development during the breeding season. And so some of our females started developing these nuptial pads. They started exhibiting amplexic behaviors. And so they were amplexing other individuals in the colony. And their ultrasounds started looking more on the side of more reminiscent of a male ultrasound image. And that's because there was no longer any follicles. And when we looked back through the history of these animals, a lot of them had actually bred as females. And when we looked at the results from our mass spectrometry, we were able to observe that the females had very different levels of hormones across testosterone and a couple of the estrogens, which is the two main sort of characteristic, the sex steroids that we picked to look at. And these females, which we call the nuptial pad females, they actually were more reminiscent of, their hormone profiles were more reminiscent of the males than the females, even though some of them had a history of breeding naturally earlier in life. So this was quite an interesting phenomenon that occurred. As you can see in that photo, you can see the nuptial pad outlined by the yellow circle. That's a female. And opportunistically, as some of these animals started to die from old age and just any natural course, our pathologists, when doing their necropsies, noted that some of them are actually showed signs of hermaphroditism. What we have in A is basically a nuptial pad female that showed that was a hermaphrodite and had a fully developed seminiferous tubules with sperm in them, but also had this like ovarian tissue with degenerating follicles and an active oviduct. So these are really interesting things and things that we would have noted superficially on the outside, but had we not had these reproductive technologies to actually go in further, we wouldn't have noted. And so when we start to see these things, it's also important for us as captive managers, right, to be able to understand how to approach these things and understand that things may happen within our captive population that we can't account for. And so if animals aren't breeding, it may actually have some underlying causative issues that have nothing to do with us giving them hormones and trying to artificially stimulate them to breed. So this is a really cool little paper and it's available. And if you'd like to read it, either Leah Jacobs, who I know is on the call now, or myself are happy to give you a copy. It's a really good communication and worth keeping in mind when you're working with some of these animals if you start to see things occur in your populations. And with that, I think it's all done. Thank you. If you guys have any questions, anybody wants to ask a question, we are here to answer it if we can.